Let's talk about the abnormal forms of hemoglobin and the associated hemoglobinopathies now. So, what causes the formation of abnormal hemoglobin? So, the reason is mutation in the genes coding for globin. Now, we know there are two forms of globins present in hemoglobin, alpha and beta. So, what genes code for alpha and beta? So, alpha is coded by chromosome 16. Now, chromosome 16, each chromosome 16 has two genes present on it which code for alpha. And we know we have two chromosome 16s, hence 2 into 2 makes it 4 genes which code for alpha. Next is beta globin. Now, each chromosome 11 has one gene which codes for beta globin. So, that makes it two genes which code for beta globin. Okay, so now we know what genes code for globin chains. Now we'll take a look at the defects which result because of the formation of abnormal hemoglobin. So there can either be a defect in the amount of globin that has been formed, which result into quantitative defects. Now these quantitative defects are also called as thalassemia. Now thalassemia, depending upon which globin chain is present in abnormal numbers, it can be classified as alpha thalassemia or beta thalassemia. We'll take a look at these thalassemias in detail later on. Now about qualitative defects. So qualitative defects can be of the types HbS, which basically means sickle cell anemia. There are other forms of qualitative defects in hemoglobin like formation of HbC, HbD and HbE. So we'll first take a look at HbS, which is sickle cell anemia. We'll start by considering its genotype. So in sickle cell anemia, the genotype is HbS. So if a person has this genotype, we can conclude that the person has sickle cell anemia. But if the person has HbS, HbA as their genotype, or HbA, HbS, whichever way, as their genotype, the person is a carrier. And if the person has the genotype as HbA, that gives us the conclusion that the person is normal. So we have studied the genotype now. Now let's take a look at what exactly happens in a sickle cell anemic person. So when a person suffers from sickle cell anemia, they show certain characteristic features like lifelong hemolytic anemia. Now why does hemolysis take place? See, as the name explains, sickle cell. So there is sickling of RBCs. We know normally RBCs are supposed to be biconcave in shape, but there is sickling, so which causes a abnormal shape of the RBCs. Now since RBCs have turned into something abnormal, our body starts destroying them, which is basically hemolysis, destruction of red blood cells. So, and since there is deficiency of RBCs, it results in anemia. Hence the condition causes hemolytic anemia. Secondly, there is tissue damage. So why does this condition cause tissue damage? So this condition causes tissue damage because there is reduction in the ability to carry oxygen. So there is hypoxia at tissue level. Since there is hypoxia, it is ultimately going to cause tissue death, necrosis or tissue damage. So there's that. The next feature is increased susceptibility to infection. So the person is also susceptible to numerous infections. Now these are the drawbacks which sickle cell anemia causes. But there is one advantage that people suffering from sickle cell anemia have. Now that is resistance to malaria. Now why does this condition cause a resistance to malaria? Now and in which group of people is this commonly observed? So people that live in tropics like the Africans are resistant to malaria because many of them suffer from sickle cell anemia. So how does sickling of RBCs give an advantage to these people? So there are three reasons. The first one. So the first reason is, since there is increased hemolysis, the malarial parasite, which basically depends upon the RBCs for its survival, is not going to be stay in the body for longer, survive in the body for long. Secondly, because of the entry of the malarial parasite, there is an increase in H plus concentration. Now this H plus concentration results in increase in sickling. So again, it's going to cause increased lysis, and hence the parasite won't be able to survive for long in the body. Next is sickle cell anemia. The people who have sickle cell anemia, their RBCs have high concentrations of potassium, and these high concentrations of potassium are not favorable at all for the malarial parasite. So basically, all of these factors are going to have a cumulative effect and be unfavorable for the survival of the malarial parasite. Okay, so now we'll look at the condition of the hemoglobin in a normal person and in a sickle cell anemic person. So if we look at the deoxyhemoglobin of a normal person, we'll see presence of receptor sites like these sites. This one and this one is the receptor site. Now this is the deoxyhemoglobin of a normal person, so deoxy Hba. Similarly, there are going to be receptor sites present in a sickle cell anemic person as well, but there is going to be an addition to this hemoglobin, to this deoxyhemoglobin. There are going to be sticky patches present here. So this is sticky patch 1, this is sticky patch 2. So there's going to be presence of two sticky patches in the deoxyhemoglobin of a sickle cell anemic person. Now, whenever the hemoglobin gets oxygenated, these sites, these receptor sites, are going to get covered. So this is going to happen in both a normal person as well as a sickle cell anemic person. But in case of deoxyhemoglobin HbS, or in the deoxyhemoglobin of a sickle cell anemic person, these sites are going to stay open. So what's going to happen is that this sticky patch is present on one hemoglobin. Say this is one deoxyhemoglobin. It has sticky patches, and this is another deoxyhemoglobin. Now this is the receptor site, another receptor site. It has sticky patches. Now sticky patch of this hemoglobin molecule is going to get attached to the receptor site of the other hemoglobin molecule. Now this situation is going to happen all throughout the hemoglobin molecule. So now this is going to result in the formation of a sort of a polymer of deoxy HbS. So this polymerization ultimately causes the RBC to become rigid. So the RBCs become rigid, and this rigidity ultimately results in the formation of an RBC which is sickle shaped. So this is what's exactly happening in a sickle cell RBC. Okay, now we'll look at what abnormality is taking place at the amino acid level. So in a person with sickle cell anemia, in beta chain at the sixth position, normally a person should have glutamate, but in a sickle cell anemic person, glutamate will get replaced by valine. Now, glutamate is a polar amino acid, while valine is a non-polar amino acid. So, we know non-polar substances will be less soluble. So, this decreased solubility will cause a decrease in the oxygen carrying capacity. So now we know why there is a reduction in the oxygen carrying capacity, because valine is nonpolar. We also saw what is actually causing sickling of the cells. That is because of presence of the sticky patches in deoxyhemoglobin. So let me make that clear that in deoxy HbS there is going to be sickling, but when HbS is oxygenated, or when it's oxy HbS, the receptor sites will be covered. So even though there is a presence of sticky patches, they won't be able to bind anywhere. So there won't be any sickling that will take place. So if we keep the cells of a sickle cell anemic person oxygenated, so the condition won't be resulting into sickle shaped RBCs. So there's that. Now, how do we diagnose whether a person has sickle cell anemia? So the very obvious option is obviously studying their genotype. So we can study their genotype by karyotyping and other methods. Next, we can also go for sickling tests, in which we'll be taking a smear of the blood and we'll check whether sickling takes place. Also, we can go for electrophoresis. Now, how will we detect whether the person has sickle cell anemia or not? So, we just need to remember this as a fact that in electrophoresis, Hba will travel at a faster rate than Hbs towards the anode. So, Hba travels faster towards the anode than Hbs. So, if we take a look at the representation after performing electrophoresis, so if a person has HbS, we'll be seeing a representation before Hba band forms, and we'll be getting to know that the person might have sickle cell anemia. Okay, now how will we manage this condition? So to manage this condition, there are two things that are commonly done. First of all, obviously, the person will require many blood transfusions. So numerous blood transfusions will be required. Also, people are administered sodium cyanate. Now, what does sodium cyanate do? So sodium cyanate increases the oxyhemoglobin formation. Now, we've seen earlier that if there is presence of oxy HbS, sickling won't be taking place. So this ensures that there are no sickle shaped RBCs, or there is a reduction in the amount of sickle shaped RBCs. Also, in recent times, certain gene therapy techniques which use the method of replacing HbS by HbF are being tried and are in the pipeline, are being experimented. But this, as of now, is not a very sure shot technique of treating this condition. Alright, so now we have covered sickle cell anemia. Now let's discuss a little bit about the other abnormal qualitative conditions which result in abnormal hemoglobin. So we saw that there's formation of three other types of hemoglobins: HbC, HbD, and HbE. So let's talk about HbC first. So HbC is also called as Cooley's hemoglobin. Now what happens here is, at 6th position in beta chain, the glutamate is replaced by lysine. Now this condition shows the symptoms of mild hemolytic anemia. That's all we need to know about HbC. Then there is HbD. HbD is a condition in which at the 121st position, again in the beta chain, normally it has to have glutamate present, but your glutamate is replaced by glutamine. That is all about HBD. Now HBE. Now the important thing about HBE is the presence of HBE is the second most common condition after HBS in qualitative abnormalities of hemoglobin. So HBE, in case of HBE, at the 26th position again of the beta chain, there is replacement of lysine. Now there is replacement of glutamate by lysine. So glutamate gets replaced by lysine in case of HBE. Now, just as a fact, just try to remember this order, that HbA travels faster towards the anode than HbS, and HbS travels faster than HbC. Also, HbS and HbD travel at the same rate towards the anode. So we can basically use this order in detecting the type of hemoglobin or detecting the type of abnormal hemoglobin present in the person. So there's that. So now let's move on towards the quantitative abnormalities of hemoglobin. So the quantitative abnormalities, as we said, it's called thalassemia. So the quantitative abnormality of hemoglobin is called thalassemia. Now, thalassemia can be because of reduction in the quantity of alpha globin or reduction in the quantity of beta globin. So, obviously, if alpha globin quantity is affected, so there's going to be some defect in chromosome 16. If beta globin's quantity is affected, there's going to be a defect in chromosome 11. So, let's first take a look at alpha thalassemia. Okay, so alpha thalassemia can be of 4 types. Since we know that there are 4 genes and all which code for alpha, so the first abnormality can be there are only 3 genes which are coding for alpha. The second abnormality can be there are only 2 genes coding for alpha. The third abnormality can be only 1 gene is present which codes for alpha, or there are no genes present which code for alpha. So if there are 3 genes present, we call it a silent carrier. So the person is a silent carrier if they have only 3 genes. Silent carrier. So this person will have no manifestations because 3 genes can still compensate for the deficiency of 1. So this person will have no manifestations. So this person will be almost normal. Next, now if there are 2 genes present, so 2 genes will, now this condition is called as alpha thalassemia trait. So there are exactly half the number of genes present which should normally be required to be present for normal synthesis of alpha. So now 2 genes get slightly burdenized now. So we'll have minor anemia. So there's presence of minor anemia. Then we'll move on to 1 gene present. So if there's only 1 gene which codes for alpha globin, the condition is also called as HbH condition, hemoglobin H condition. Now this person, now the load on the gene is further increasing. So this person will be moderately anemic. Finally, if the person has 0 alpha globins, we refer to this condition as hydrops fetalis. And this person or this condition results in death almost after birth, very close to the post-birth situation. The person dies. So the person dies post-birth. So we've covered alpha thalassemia now. So now let's take a look at beta thalassemia. So we've seen that beta thalassemia, that beta globin is coded by 2 genes, each 1 gene each on both of the chromosome 11. So if there is presence normally there should be 2 genes present for beta globin. But if there is a condition in which there is only 1 gene which codes for beta globin, so this condition will be called as presence of beta thalassemia trait, or this condition is also called as beta thalassemia minor. Beta thal minor. Now this person would be asymptomatic if there is only one beta globin, 1 gene coding for beta globin. So this person can still survive with 1 gene. But if there is presence of 0 genes which code for beta globin, the person won't die immediately after death. But because the reason is beta globin is not synthesized at birth. Beta globin synthesis starts once the baby is born. So eventually, when the body will start facing deficiency of beta globin because of lack of its synthesis, since there are 0 genes coding for beta globin, the person will start facing difficulties, and eventually this is going to result into death. So that is all, beta thalassemia. So what we've seen mainly is there are hemoglobinopathies, which are basically conditions wherein there is abnormal hemoglobin synthesis, which results into some difficult conditions for the person. So now these hemoglobinopathies can either be because of the poor quality of alpha and or beta globin chains, or the poor quantity of either of the globin chains. Now poor quantity can either be because of alpha thalassemia or beta thalassemia. Now they have further more types depending upon the number of genes absent. Qualitative is either HbS, sickle cell anemia, or it can be HbC, HbD, or HbE. So that's all.